Hey y'all, my name is Prashanth Kumar, or PK, and I'm an MCAT tutor at Shemmassian Academic Consulting. Today, we're going to be doing an MCAT Biochem passage walkthrough. You should be taking roughly 8 minutes on this passage, but today we're going to be going more in depth so you can see exactly what's going on in my head as we take on this passage. Let's get it. Alright, great. So I've gone ahead and put the passage on the left and the questions we're going to go through on the right. So feel free to take a moment, pause the video, take your time, and come back to us. If not, let's get right into it. So let's talk about strategy first. I like to maintain the same strategy going into every single passage that I come across. The reason for this is it gives me something to come to regardless of whether the passage is easy or it's hard, right? We want to be grounded, getting ready to go into the test, even if the passage is something we've never seen before. And so for me, that strategy looks like reading a paragraph, writing a short 3 to 5 word summary, and then going to the next paragraph, and on and on and on. The reason for this is I think it provides a little bit of accountability, making sure I understand exactly what's going on in that paragraph before going on to the next. Oftentimes, I just start reading really emptily, not understanding anything, just because I was worried about time. And I'd get to the end of the passage and be like, what just happened? Doing this little check-in of 3 to 5 words, make sure I understand what's going on in a paragraph. And if I don't understand it, I can just read that paragraph really quickly again, rather than doing the entire passage over again. So the next thing I want to add to my strategy is when we come across figures, I don't want to waste too much time on them. The reason for this is we don't know what the MCAT's actually going to ask about these figures, right? And it doesn't make sense to waste any time, especially if the MCAT doesn't ask any questions about a figure, right? So let's get a big picture idea, maybe skim it for a second or two, and then go on to the next paragraph and come back when we actually need to. All right, with that, we're ready to get into the paragraph itself. Nuclear estrogen receptor 2, ERB2, suppresses tumor growth and modulates cancer cell proliferation in breast cancer. Loss of ERB2 is shown to correlate with early stages of ductal and breast tumors, though the specific mechanism is largely unknown. Researchers hypothesized that identifying and characterizing multi-protein complexes involved in the ERB2 function might identify the molecular basis for the role of protein in BC. So the question I ask myself every single time after I read a paragraph is, what's happening? Pretty simple summary, 3 to 5 words, symbols, arrows, encouraged, right? And so for me, that just looks like ERB2 inhibits tumor, right? And tumor growth modulates cancer cell proliferation in breast cancer, right? All of these are details we can come back to if we need. But right now, we're not going to focus too much on it. We just want to have a big picture understanding, not get caught up in the details, and go on to the next paragraph. Using interaction proteomics, which combine native protein complex purification and mass spectroscopy, it was determined that ERB2 interacts with other proteins in BC cells, and this interaction is mediated by one or more RNAs. Okay, so again, question is, what is happening, right? We see this ERB2 interacting with proteins in BC cells, right? And so that's the basic main idea. And we also see that RNAs are mediating this, right? There's this involvement of RNAs in this interaction between ERB2 and proteins in BC cells, right? Great. And right away, we're ready to go on to the next paragraph. To probe the effect of RNA on ERB2 complex formation, researchers treated a nuclear extract containing ERB2 and its putative binding partners with RNAs. The results from the experiment are shown in figure 1. So exactly, we have a figure, this sort of a lead-in passage looking at what's happening in the figure, and what is happening. We're just looking at ERB2 with RNAs, right? And we can look at the figure for a second. We see RNAs is changing, but it's a Western blot of ERB2. That's the basic idea. And so right away, we don't want to spend too much time on this. Let's go on to the next paragraph. Next, the researchers analyzed ERB2 complex formation with putative binding partner AGO2 in the absence and presence of RNAs. The results are shown in Figure 2. The researchers analyzed the nuclear extract, the amount bound AGO2, and 2 TeV IgG cephalose column eluents using Western blot for AGO2, another figure, right? This is the same exact structure as the previous paragraph, where we saw a paragraph introducing a figure, same thing, paragraph introducing a figure. So what's happening? Well, we have ERB2 and interacting with AGO2, right? That's what it's saying. We're checking out the interaction of the binding partner with ERB2. And how is this modulated? Well, we're looking at how RNAs modulates this, right? We're looking at what happens as we change this RNAs in the expression. And if we look at the figure again, right, we see it's a Western blot analysis of AGO2. That's all we really need to focus on right now. We'll come back to all the details later, but with that, we've done it. We are ready to go on to questions. I'm going to put the question right here. Which of the following most likely contributes the greatest amount of stability to the ERB2 AGO2 interaction? A. Disulfide bridges. B. Hydrogen bonding. C. Hydrophobic effect. D. Salt bridge. All right. So with this question, we have to understand what exactly we're looking for, right? We're looking at this ERB AGO2 interaction. And what paragraph focused in on that? Well, it was this final paragraph, right? But we don't know it needs to go too into depth, right? It's just asking what's most important for the stability of this interaction. So that just requires understanding what ERB2 and AGO2 are, right? And we see ERB2 is a protein, right? And AGO2 is also a protein. Great. And the reason we can see this is by looking at the Western blots, right? Figure 1 is a Western blot of ERB2, and Figure 2 is a Western blot of AGO2. And there's a nice little mnemonic that I'll write in blue here to help you remember what each of the blotting systems are for. So snowdrop. S is associated with southern blot, and that is associated with DNA. N is northern, and that's associated with RNA. O is sort of a placeholder, and western is associated with P, which is protein. So we know both of these are proteins because we're dealing with Western blots. So now we're looking at what is most important in a protein-protein interaction, right? And this is something we should know from our content review. It should be the hydrophobic effect, right? The hydrophobic effect is very, very important in maintaining stability between proteins. Hydrogen bonding, disulfide bridges, even salt bridges are all associated with that protein amino acid relationship. There might be minute forms of salt bridges or hydrogen bonding stabilizing proteins, but it's really, really, really protein hydrophobic. So that's what we should focus in on. So we can cross out the other answer choices, and we're left with C, and we're 1 for 1. Great work. And we're on to question number 2. Researchers decide to purify ERB2 using a nickel column. They most likely prepare ERB2 with a C-terminal His6 tag, B, N-terminal ALA6 tag, C, C-terminal GLU8 tag, D, N-terminal Val8 tag. Okay. So here we have a pretty challenging question, right? It's talking about nickel columns, which is a type of experimental technique that we should have studied in our content phase. But we're looking at these tags, right? And we should remember something about this, but we're not too sure whether it's C-terminal or N-terminal. Well, the first thing is we actually don't need to look at it. This is something made to throw us off a little bit. It says C-terminal, N-terminal, C-terminal, N-terminal, whereas all of the amino acid residues are different. So we don't really need to focus on what terminal they are so much as we need to focus on exactly what the tag is, what amino acid it's based off of, right? And we know it's associated with a nickel column. And from our content review phase, The nickel column is associated with one specific type of tag, and that is the histidine tag. So this is a standalone question that's pretty much based on understanding something from a previous sort of content review phase. Pretty challenging to get from this passage because we don't really talk about nickel columns or tags anywhere in there. The reason for this is that the histidine tag actually chelates the nickel of the column. And so there's a great article on the Shemmassian website. Feel free to check it out to learn more about it. But the histidine tag is probably the most commonly used tag for these nickel columns. And these can occur on either the C or N terminal. So this question is made to throw you off a little bit with trying to pick which terminal, but if you realize that the amino acids are all different, you can just focus in on that as opposed to focusing in on whether it's C, N before coming back to the question. All right, challenging question, but we're 2 for 2. Let's get right to the next question. 3. Okay. To interpret the results of figure 1, the researchers must assume all the following except A, equal concentrations of RNAs were used in the RNase plus lanes, B, equal concentrations of nuclear extract were loaded in all of the lanes, C, equal amounts of ERB in complex with other proteins form in all the lanes after RNase treatment, and D, the antibody for ERB is specific to the protein at a site distinct from protein interaction sites. All right, so we're looking at figure 1, right? This is an example of a question that makes us go back to the figure we're looking at. And so we have a Western blot with RNAs, negative, 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 positive, 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 different sort of trials with RNAs or without RNAs, right? And so what is necessary for this is a great question to ask, right? Equal concentrations of RNAs were used in the RNase plus lanes, right? If this is an experimental technique and we're changing one variable, the presence of RNAs, right? We want to make sure that we keep the amount controlled, or else our sort of lanes will look vastly different from one another, right? And so this is an example of using the amount, equal concentrations as a control to make sure that we know how much we're changing, we know what it's going to look like if we change it. So A is going to be eliminated. B, equal concentrations of nuclear extract were loaded in all of the lanes, right? This is another important control, right? We want to make sure we have an equal amount of this extract so we can see the banding patterns, right? We want to make sure that any variation in banding patterns is not due to us putting in the wrong amount or a different amount of extract in each of the columns, right? Perhaps we see something a little thinner, right? How do we know it's not because of having a little less extract, right? We want to make sure that we have equal amounts of extract loaded in all of the lanes. So we're going to eliminate that. So those A and B are pretty good controls that we know, like in any standard experiment, we want to keep those the same so we're not confusing, we're not confounding our outcome variables, basically. C, equal amounts of ERB in complex with other proteins form in all of the lanes after RNase treatment, right? And so this is something that we have to think about, right? This actually doesn't sound that true. Equal amounts of ERB in complex with other proteins? Well, we see different banding patterns in every single lane, right? Well, they might be between the negative and positive lanes too, right? After RNase treatment, they're not the same banding patterns. And so this is something we're going to come back to because we see, okay, we're not going to see exactly the same ERB in complex with other proteins, varying with all the lanes. So let's just be careful of that, and we'll keep C on our answer choice. D, the antibody for ERB is specific to the protein at a site distinct from protein interaction sites. Okay, this one is actually very true, right? We need to keep these binding sites distinct, or else what if another protein binds and confounds our variable again, right? We need to make sure that another protein cannot bind to our binding site and change our results in a different way. And so D is also a necessity to maintain for Western blotting. So we're left with C, equal amounts of ERB in complex with other proteins form in all the lanes after RNase treatment. Great. So right away, this is a great example of a question that makes sure that we understand experimental design, variables that can confound, as well as sort of how we maintain controls to maintain the highest quality experiment possible. So 3, good work. And we're on to question number 4. Why do researchers include the input lanes in both RNA's conditions in figure 2? A, to determine the concentration of AgO2 before the experiment. B, to ensure that equal amounts of AgO2 were present before the experimental treatment. C, to measure the amount of ERB2 complex without RNAs. D, to increase the likelihood of binding events for later conditions. All right, so here we have something that points us to figure 2, so exactly the same thing. We have to go back to the figure. And it's asking us to focus in on these input lanes, right? And so which lane is that? That's the first lane right here. And so they're asking, why do we include the input lane? And this is a part of the experimental technique that we're going to have to know. The input lane is sort of this control that we focus in on to make sure that we have the exact same amount of something before our experimental treatment. Like I said, we don't want different variables confounding our experimental results. If we had too much of our extract or something initially, right, then we'd expect more dense banding patterns in the future. So by maintaining an equal amount of this sort of input column, we make sure that our results are comparable, right? We can maintain that everything else is kept the same, and we can change this one variable and see what the results are. And so that aligns best with one of these answer choices, and that's answer choice B, right? To ensure that equal amounts of AgO2 were present before the experimental treatment. And by having this sort of maintained, we can see exactly what is being changed after the experimental treatment. To determine the concentration of AgO2 before the experiment doesn't really make sense. Western blots don't provide information about concentration, so we're going to scratch that out. C, to measure the amount of ERB2 complex without RNAs. AgO2 is being worked with, so not ERB2, so we should just focus in on that, right? We see that AgO2 down there, and so we can eliminate that. D, to increase the likelihood of binding events for later conditions. Doesn't have to do anything with binding events, so we're going to cross that out. This one's a great question. Make sure we understand exactly why we're adding these controls in the beginning. And so we have done 4 for 4, and we're on to question 5. Oops. There we go. Which of the following conclusions is best supported by figure 2? A, an RNA molecule plays a role in the AgO2-ERB2 interaction. B, ERB2 is not expressed when RNase is present. C, AgO2 binds ERB2 more tightly in the absence of RNA. And D, AgO2 is upregulated when RNase is present. All right, here's another example where we have to go back to this figure 2. And so what is happening, right? We're saying we're adding this RNase from one blot to the next, right? And so what is changing? Well, we see that there's like dense blotting here, whereas the blotting here is much lighter. So we can just write lighter and darker, okay? Just to focus in on it. What does that mean, right? What does a darker band mean, and what does a lighter band mean? Well, the darker the band is, that means there's more quantity of this protein, okay? So increased expression with this, with this. So without RNase has more expression of this protein, whereas with it has less. So that's the big picture understanding. Let's also make sure we understand what RNase is. Ribonuclease. Anything with A's has to do with degrading, right? And so this is actually something that degrades RNA, right? Degrades RNA. So this is a very important idea, right? In the presence of RNAs, right, we would expect increased degradation of RNA. Whereas without RNAs, we'd have less degradation of RNAs. So now, let's go to our answer choices, right? A, an RNA molecule plays a role in the AgO2-ERB2 interaction, right? So we can think of this as without RNA and with RNA. So an RNA molecule plays a role in the AgO2-ERB. If we get rid of RNA, are we seeing a change in the banding pattern? And the answer is yes, right? So if we lose this RNA, with this RNA being added, we expect to see lower banding. So right now, A is looking like a great answer, so I'm just going to put a dash there. B, ERB2 is not expressed when RNase is present. Well, this plot is only looking at the expression of AGO2, so this is not being looked at, so we can cross that out like we said in the same answer choice last question. C, AGO2 binds ERB2 more tightly in the absence of RNA. So let's think about this. This is the exact opposite of what A is saying, right? It's saying AGO2 binds ERB2 more tightly in the absence of RNA, or without. So if that were true, we'd expect darker banding in the second plot as opposed to the first plot, which is not true. So we can cross that out. D, AGO2 is upregulated when RNase is present. We don't really see that. The input level is the same, so with or without RNase, AGO2 is not going to be upregulated. So we can cross that out as well. So right away, we're sticking with answer choice A, and this is an important idea, right? We needed to understand exactly what RNase is. What is being changed from that first plot to that second Western blot? And increased RNase, we see it means that there's less RNA present. And so with that, we're able to tell the difference between that first plot and that second plot, and we get our answer choice right. We're 5 for 5. Great work, team. All right, great work, everyone. We crushed another biochem passage dealing with experimental techniques, Western blotting, and so much more. So kudos to you to getting it done. If you like this, give us a like down below and subscribe to our channel. And feel free to comment exactly what you want next so we can tailor these sessions for you. Feel free to check out our 528 series from Vikram for all the test-taking strategies that you need to maximize your score. And additionally, sign up for our MCAT question of the day so you can get every last bit of studying done before test day. Let's get it, and happy studying.